Okay, our next speaker is Saphira Bloom-Quinn. Saphira is also from the Research Centre for Ecosystem Resilience and Botanic Gardens in Sydney. She received her joint MSc from Uppsala University, LMU Munich in evolutionary biology, and is working at the centre at the moment and applying population genomic data to pertinent restoration questions. And today Saphira is going to talk on using landscape genomics to uncover genetic patterns in Banksia marginata, implications for restoration strategies. Thanks, sir.
Just gonna check this laser works, yes. Awesome. Can you hear me fine? Yeah. So I'm Saphira. I'm also from the RESA, and as mentioned, I'm presenting the results of our landscape genomics study on Banksia marginata today and discussing the implications of that work on seed bank collections, seed production areas, and also restoration work. And I'd like to acknowledge today that I am on Jagera, Giabal and Jarowa country and pay my respects to elders past and present. Also like to acknowledge the Gadigal people of the Eora Nation where I live and work, as well as the numerous traditional custodians who have taken care of country on which the collections for this study were made. So Banksia marginata, known as the silver Banksia for the silver underside of its leaves, is one of the most morphologically variable Banksias, and it has a wide natural distribution from the north of New South Wales down to Tassie and across to South Australia. And we know anecdotally that its range has reduced since colonisation. So historic range estimates have been made by Dr. Steve Sinclair from the Arthur Rylah Institute and presented, and he used a variety of different records to estimate the historic range of Banksia marginata in the Victorian volcanic plains, and he speculated on a number of causes for the decline. However, I'm not aware of any other studies that systematically map the decline of Banksia marginata. So I've plotted here herbarium records since 1980 with the more recent records in darker reds to give a little bit of insight for where the more recent losses may have occurred, and they seem to be concentrated on either side of the Victorian volcanic plains, also southwestern Tassie here. So despite concern about this decline, there have been very few genetic studies of Banksia marginata. I know only of two, both from Victoria, one in based in seed production areas and another of wild populations. And so without kind of a more wide-spanning genetic study, we don't know what the main genetic groups of Banksia marginata are. We don't know how genetic connectivity varies across the landscape. We don't know what the patterns of genetic diversity are across the landscape. And without this information, we struggle to make genetically representative seed bank collections and also to make genetically effective seed production areas and restoration work. So with this in mind today, I'm presenting the results of a study that's been many years in the making. We've worked with numerous collectors over the years to sample, that's in the red spots here, multiple individuals. So after filtering for minimum site numbers, we have 355 individuals across 61 sites, which is pretty cool. And that has been sequenced using diversity arrays technology, and we have just shy of 3,000 quality variable genetic sites to make our inferences from. So this first analysis here, this splits tree, which shows the data as a phylogenetic network, we can see that New South Wales are these individuals with the red colours, and we can see that there are two groups. There's this smaller subset made up just of New South Wales samples, and then there's other New South Wales samples that are in a bigger clade along with the rest of the range of Banksia marginata. Next is this principal components analysis, which reduces down the dimensions of the data into its main components, and we can see along the main axis of variation, we again have most of it's explained by this one cluster, this subset of New South Wales samples, and then this other cluster, which is made up of everything else. Next, this is a LEA analysis, which helps tell us about population structure, ancestry, and admixture. And the results shown here are for two ancestral clusters. And the LEA analysis estimates that this first ancestral cluster begins in northern New South Wales and is the dominant ancestral group around Sydney and the Blue Mountains. And the second ancestral group is everything else. So if we go back to the splits tree and PCA here, and we colour the tips by those two ancestral groups, we find really strong support for the same two groups coming up again and again, with the grey individuals here being individuals that are highly admixed or have mixed ancestry. So identified two distinct genetic groups, and a representative seed collection should represent both of those. And restoration work in the Sydney region should source seed from the appropriate genetic group. And there's limited evidence for strong population structure across the rest of the range, even though this encompasses several thousand kilometres. So looking at genetic connectivity across the landscape, for this I'm first looking at FST, which is a measure of divergence between sites, and in that way is an indirect measure for gene flow. And we find that sites from northern New South Wales are very divergent from all other sites. Then we find that sites from the Sydney area have moderate levels of divergence with everything else. And we find that sites from the main clade, despite the fact that they represent 1,500 kilometres of distance, are not very divergent from each other. So if I plot FST here along the y-axis, along with the pairwise distance on the x-axis, we find two things. One, as you expect, if your sites are close together, they are more similar than if they are far apart. This pattern is called isolation by distance. However, considering the large range here, up to like 1,200 kilometres here, it's pretty shallow. And secondly, we find that for the same distance, if your sites are from the same group, same genetic group, so that's in blue, they are more similar to each other than for the same distance if those two sites are from two different genetic groups. So the northern New South Wales sites are highly divergent from all other sites. This suggests there's possible barriers to gene flow or that it's a disjunct population. Gene flow appears highly restricted, even between highly proximal sites in the sort of Blue Mountains area. So again, restoration work should source genetically informed seed to ensure that they're reproductively compatible. And sites across the range of the main clade show weak patterns of isolation by distance, which suggests that gene flow isn't limited across the range. However, with the caveat that the range contraction might be too recent to have left a signature on this particular metric. Moving on to genetic diversity, here I'm looking at site heterozygosity, which tells us something about how often variants are carried in the heterozygous state, so by the same individual at a site, and so it's a measure of genetic diversity. These measures are sensitive to population structure, so they've been performed per genetic group, and we find that heterozygosity is moderately high across the range at around 0.17. Next, I'm looking at FIS. This measures how much genetic diversity is contained within an individual compared to its site, and so estimates inbreeding. And we find that FIS estimates are neutral to slightly positive across the range, which indicates that low levels of inbreeding may be occurring. So levels of heterozygosity are moderately high. This suggests an outcrossing mating system. This is important because seed production areas should therefore carefully manage inbreeding, and this means monitoring maternal lines carefully. Low levels of inbreeding may be occurring in wild populations, potentially due to range contraction. So moving on to how Banksia marginata is represented in our seed bank collections. So we queried 4 different seed banks, and they reported holding between 0 and 9 accessions of Banksia marginata with an average accession age of 28 years. Those accessions are shown in the grey diamonds in this plot here. So if we expand those accessions out, 5 of the accessions probably represent that smaller Sydney bioregion genetic group, and 7 of the accessions probably represent the main genetic group. However, 3 of these accessions were collected within 5 kilometres of another accession and therefore probably don't contribute anything novel genetically. So if we remove those and then take a look at the age of the accessions, even though Banksia seeds are known to be long-lived and probably viable for years to decades, if we even apply a quite not a very harsh threshold of 2 decades, we're actually only left with 2 accessions in our seed banks for Banksia marginata, and they're both from the main genetic group. So I used OpGen site to get a little bit of an estimate for what those 2 accessions might actually be holding from a genetic point of view. And so with OpGen site, I can estimate the proportion of common and rare variants that we would expect to get for randomly sampled sites. So for those 2 accessions, if they represent multiple maternal lines, we would approximately have captured 80% of the common variants and 30% of the rare variants, which is very encouraging for that particular genetic group. And then kind of moving on to where to go from here. So with OpGen site, we're also able to select optimized sites, so for the same number of sites, 2, 3, or 4 sites. And these optimal sites are significantly better at capturing rare variants than random sites are. And these rare variants may be of interest because they may be important for future climate scenarios, for example. So this is the recommendation that OpGen site suggests for an optimal collection for the main genetic group. And this NAGI group is already represented in our seed banks, which is really great. But those are the other recommendations for a quality collection. And for the Sydney bioregion group, where we currently don't have any accessions under 2 decades old, these would be the 4 sites that are recommended for an optimal site. But overall, the kind of patterns we see is that sampling more than 3 sites doesn't capture significantly more common variants, but obviously we want to build a little bit of robustness into the collection because not everything is going to survive. And then sampling 6 or more sites is recommended to capture more than 90% of the rare variants. So depending on what your goal is for making a conservation seed collection, you kind of need to keep these different things in mind. You do need to collect from more sites to capture more of the rare variants. So this is a promising starting point. We don't have to collect from like a crazy amount of sites to be able to capture quite a lot of the common variants and rare variants. So it's a really good starting point, but we want the collections to be robust, obviously, because not all of the seeds are going to survive. So putting it all together, we've identified 2 main genetic groups, but seed collections currently only have recent accessions for 1 genetic group. There's limited evidence for strong population structure across most of the range, spanning from New South Wales all the way to South Australia. There's moderate divergence between even very proximal genetic groups in the Sydney Blue Mountains area, which indicates they may not be reproducing with each other, and so restoration work should source genetically informed seed. Heterozygosity is moderate, suggesting an outcrossing mating system. Seed production areas should carefully manage inbreeding for this species. And conservation seed collections can capture common variants within just a few sites of good sampling, so representing multiple maternal lines, but more sites are required if you want to capture all of or nearly all of the rare variants. With that, I'd like to sincerely thank the very many collectors who have worked on this project.